Okay, my name is, can you hear me now?
Yeah, I can hear you.
Okay, my name is, thanks for having me. This is only my first experience demonstrating with Zoom, so I hope it works. But my name is Michael McWilliams. I'm a forest pathologist with the U.S. Forest Service. I've been doing pathology for more than 40 years, and I moved to LeGrand in 2013 and inherited the humongous fungus. Some friends of mine had did the original work on this, as I'll explain later, but uh, it happens to be in my area, and there is constantly interest in the biggest living organism on Earth, as you might expect. So I've gotten to be on several TV shows about this. I've gotten to host the Washington Post photographer in the woods and several other things. So I'm glad to be able to tell you about this today because it is a story that keeps coming up. So being a forest pathologist, I'm going to talk about the humongous fungus, which of course is a root disease, but I'm not going to talk about other amazing forest fungi facts along the way. If my talk goes entirely too long, I hope to, I'll have the link for the OPB uh, Oregon Field Guide session on at the end, and you can look at that for yourself. But I'll just launch right in. I'm going to talk about the humongous fungus and other amazing forest fungi. This is what I'm going to talk about. I'm going to talk about the roles of fungi in the forest. I'm going to have a little bit of basic biology, talking about basidiomycetes and ascomycetes. I'm going to talk about some examples of forest diseases. And then most of my talk will be talking about the details and research on the Armillaria clone that's on the Malheur National Forest. I was at your presentation last month, and the speaker did a pretty good job about talking about some of the roles of fungi in the forest. The mycorrhizal fungi are very interesting in that they have relationships with many plants, including trees and other plants, and it's basically the mycorrhizal fungi are culturing up the green plants. Many of them would not be on Earth if it wasn't for the fungal relationship they have with their roots. I'm also going to talk about parasites. That's what I make much of my living at, is dealing with plants or fungi that live on living plants. And I'm also going to mention saprobites. They live on dead plants, and many of the fungi you've been talking about picking and eating are either mycorrhizal or saprophytes. So mycorrhizae, and I just got this off Wikipedia. It's believed that the development of arbuscular mycorrhizae symbiosis played a crucial role in the initial colonization by land plants. 80% of vascular plant families have arbuscular mycorrhizae, and about 2% have ectomycorrhizae.
We don't know what that means.
I know, I'm going to tell you. Arbuscular mycorrhizae, can you see my pointer? Actually
Yeah, I
penetrate.
see it now. Yeah, I see it.
Okay. They actually penetrate plant cells and form these arbuscules within the plant. And so they're called vesicular arbuscular. They actually have arbuscules that directly feed the plant cell. Ectomycorrhizae basically is a sponge sheath around roots. And you can see from this picture, the fungal network greatly expands the water collecting and nutrient collecting capacity of the plant. So a lot of plants are very dependent on mycorrhizae. Another class of fungi in the woods are saprophytes. These live on dead tissue. This one here is called red belt. It's Fomitopsis pinicola. Very common in the woods. You'll see them with nice red belts sometimes they're blacker. If it wasn't for this fungus, we'd be up to our ears in dead wood. This is a major decomposer of dead wood in the forest. Another one that I like is Polypus sulphureus, chicken of the woods. This is a saprophyte that's edible. I've made some delicious mushroom soup with this fungus. It grows on hardwoods and conifers. I've eaten it off oaks and it was nasty, but I've eaten it off hemlock and Douglas fir and it was excellent. So many of these.
Does it fruit in the spring? Does it fruit?
I've seen it in the fall. So another great realm of fungi is parasites. On the left, you'll see Armillaria ostoyae, that I'm going to be talking about through most of this talk. Another great realm of parasites is the rust. This is white pine blister rust on the right, and on the bottom is stalactiform rust on lodgepole pines. These are good. Many of the parasites are good basidiomycetes related to the guild mushrooms. So when I talk about ascomycetes and basidiomycetes, you may or may not know about these, but an ascomycete is a fungus that has 8 spores in a sac. You can see these sacs with 8 spores lined up. These are your cup fungi. These are also, the morels are ascomycetes, and it's one of the 2 major phyla of fungi. Much of what I'm going to talk about the rest of the talk are basidiomycetes. These are fungi that have a basidium, and 4 basidiospores are formed off the basidium. These then flick off with a water drop mechanism and go on to infect other plants, or sometimes other fungi. So that's it for the biology. Ascomycetes are the kind of cup fungi that Linda showed earlier. Also, these morels that I found on Wednesday are other ascomycetes. Most of the guild mushrooms and the polypores, the ones with poroid surface, are basidiomycetes. I'm going to also talk about straminophiles, which are not strictly fungi, but I did a lot of work on these, and these are major plant diseases. Phytophthora ramorum is sudden oak death. It's a big player in Southwest Oregon right now, an imported disease. We don't know from where. It didn't have a name before 2001, but this has spores in a sporangia that actually have flagella, and they swim in water. So, or they blow in the air and infect in a water film that's on the leaves. You can see all these sporangia happening around this leaf disc, and they also have long-lived chlamydospores. You can isolate them. They grow really pretty in a plate, and you can do a PCR analysis to get identities. They're kind of hard to do for morphology, but there's good DNA tests. And they kill a lot of trees. They spread in wind, and there's one in Port Orford cedar that spreads in mud and water, and straminophiles can kill a lot of trees. This is the same genus as Irish potato blight, the potato famine. So it's a big player, but it's not strictly a fungus, so I'll go back to regular fungi. So there's kind of 3 big classes of diseases we talk about, one of which is needle diseases. These tend to be ascomycetes, and they cause, especially in wet springs, they cause damage on needles of various conifers. You see here in this, this is a Douglas fir needle. These are the stomata, the breathing pores on the underside. And you can see when the fungus is here, it plugs up every stomata. Now you think, okay, so we're plugging up stomata. Well, it causes enough damage that you can see it from the air. And on the right, you see an aerial survey from 2019 of stands affected by Swiss needlecast on the coast. This has become a big problem in Douglas fir plantations that are planted in places that used to be much more mixed forest with hemlock and Sitka spruce. Now it's pure Douglas fir, and these needle diseases can roar. Also, you tend to see needle diseases with plantings of trees that are off-site, meaning the seeds came from someplace else and are not locally adapted. And there's some native needle diseases that I call flash in the pans. They cause some damage, but they usually don't kill trees. Heart rots are another class of forest disease. They rot the heartwood, which is actually dead wood. So in a way, these are sort of saprophytes, although the tree depends on the central column of wood for support. So if too much of that column rots, the trees can fail mechanically, even though the fungus isn't actually killing the tree. It causes it to weaken to where it falls over. Some of these are very important in hazard tree campground situations. The one on the left is, Fomitopsis pinei. The one on the right is called Indian paint fungus, Echinodontum tinctorum. If you see two conchs like this, there's probably 35 feet of rotten wood in the middle of that tree. It isn't going to kill the tree, but it may make the tree break. And the decay in some heart rots, this is quinine conch. You can see there can be very little live wood left, and the whole interior is just rotten. This is the decay caused by ET, or Indian paint fungus. It's called red-brown stringy rot. This is called white speck. This is the one caused by this fungus, pinei. So heart rots are very important in structural decay and also degrade. If you're trying to cut trees and make boards out of them, these are not going to make very good boards, right? So many foresters learn early on to recognize the conchs. The conchs, meaning the fruiting bodies like this and like this, caused by certain fungi, because they know there's degrade and decay associated with them. So now we get to the big players, the root diseases. These are fungi that tend to be restricted to the root and just the first few feet of a tree. They usually don't go very far up the bole, but they cause major structural defect, and they can kill the tree because they not only are eating dead wood, but they're actually killing live wood. And you can see this is Phlinus wierei. It has a fruiting body that's just a brown crust with pores. You almost see nothing. This is a big player, Heterobasidium nanosum. I'll have a picture later, but this makes nice big conchs, but they're inside rotten stumps. You rarely see them on the outside of trees, but they're on the inside of rotten stumps. Here's our favorite Armillaria, ostoyae, that I'm going to be talking about. It also causes a root decay, but it makes regular mushrooms. And this is one common in spruce. It doesn't kill the tree, but it causes them to be very weak. And you can see with that little bit of sound wood left, the tree is not going to be very strong. So now I'm going to talk about the humongous fungus. This is this is the original paper that established the size of this fungus. It was in the Canadian Journal of Forest Research. Brennan Ferguson has my position in Wenatchee. He's a forest pathologist for the Forest Service. Tina Dreisbach is now actually a naturopath, but she was a forest pathologist. Catherine Parks and Greg Phillip both worked in LeGrand, where I am now. And Craig Schmidt was my predecessor in my position as a forest health protection pathologist. So it's a bunch of friends of mine that actually wrote this original paper. This Armillaria ostoyae makes wonderful mushrooms. They're called honey mushrooms, and they're quite edible. However, as your speaker last some people are sort of allergic to them. They're not poisonous, but they are disagreeable and give you GI upset. But if you're one of the 7 people that can eat them successfully, they're quite tasty. This fellow here is also a forest pathologist in Bend. This is Brent Oblinger, and I offered to give him this talk, and he said, "No, you do it, Mike." But this guy has worked a lot on your campgrounds in your local area. I just got a note that said my internet connection was unstable. Can you still hear me?
Yeah, we can still hear you. It's just a little garbled, just a little bit, but so far so good. And if it gets worse, we can start asking people to just shut down the video, their video link. Okay.
All right, so Armillaria ostoyae forms, it kills trees. It's living in the sapwood of trees. You can see the white mycelium that is just under the bark of this tree. Often the bases of trees that are killed by Armillaria are very pitchy. This is because this is a parasite. It's killing live wood. And the tree reacts by trying to wall off the infection with pitch. And so trees that are killed by this, before you even chip them, you can see that the base is very pitchy. If you chunk underneath, you'll find this white mycelium that can be quite thick. And as you see, they make mushrooms sometimes, not every year, but sometimes. And when they do make mushrooms, as in this tree, you can see they can make a lot. But more often, what you see is dead trees in the forest. You see standing dead trees. You see trees that have been tipped over because the roots were completely decayed by the fungus, so they fell over. And you may see trees that are just dying. What's interesting about this picture is many of these trees that are living are young larch. Western larch is very tolerant of this fungus. And if it gets old enough, it doesn't die at all. The trees that are most susceptible to Armillaria are the grand fir and Douglas fir. Pines are more resistant, and larch is very resistant to being killed by the fungus. So what you see is these, in the forest, is these stands that are falling apart. A lot of the grand fir and Douglas fir is dying. Other tree species and brush species are coming up in the middle. You can see there might be reproduction of young trees in the middle of a root disease center, but there's trees dying around the edge, and there's older dead trees in the middle of these root disease centers. This is basic. If this fungus is present, it makes it very difficult to successfully grow trees. Another view of a Armillaria root disease center showing the standing dead and the trees that have fallen over and are on the ground, and you've got young trees coming up in the middle. So how big is the humongous fungus? This is a slide from Alex, who I'll show you a picture of him in a minute. But a blue whale It's the biggest animal, but it's not very big. The aspen grove that's in Utah is pretty big, but it's only 106 acres. The humongous fungus, this one Armillaria clone, is 2,200 acres on the Malheur National Forest. If you add it, not only is it the biggest in size, but if you added up all the mycelial threads that are in the ground, it would be something like 200 boxcars worth of mycelium. It's there's some controversy in the pathology realm of whether we call it ostoyae or solidipes. If you know about botanical names, there's people that have found reasons for calling them both. Most pathologists tend to call it ostoyae. There are some that call it solidipes. I won't get into that controversy here, but it grows very well on media. It's easy to isolate, grows well on malt agar. And the fun thing about it is it makes these rhizomorphs, these root-like things, like crazy in the plate. I've had plates where it basically crawled out of the plate and was pushing the lid off with rhizomorphs. It also, you can grow it in culture in jars, and you can see here's a primordium of a mushroom that was right here in this jar. So it is a good edible. You can grow, it's quite easy to grow in culture. It's not an obligate parasite like the rest that need living tissue. It can also exist as a saprophyte on dead tissue. This is an important point because not only can it kill the tree, but it can hang out and live in that root system for 80 or 90 years. As new roots contact those rotten roots that are still in the ground, the fungus and the infection can spread. This is the main way it spreads, is what we call vegetatively. So it becomes a clonal organism. It's all the same genetically because it hasn't done sexual reproduction, and it just spreads through the forest from root to root to root. It also has these things called rhizomorphs, which means rhizo means root and morph. So they look like roots, but they're actually fungal structures that are enclosed in a pseudosclerotium, or this hard, you can see this is a micrograph of this hard crust with white mycelium in the middle. And it can grow in adverse conditions like dry and under the bark. It can grow short distances through soil to infect a new root. So this is a structure where it protects itself and can move from root to root because it has this hard coating that keeps it from drying out. Armillaria can form quite large root disease centers. This is a picture from a publication in Idaho where you see what we call, it's sort of ringworm on the landscape. You can see this circle. This is an Armillaria clone. There's dying trees around the edge, and there's younger trees in the middle. This is another one here. This is another one here. So it can kill a lot of trees and be a major disturbance agent on the landscape. Here's a picture of Armillaria in a pear orchard, and you can see when it gets started, it's a disease of the site. You cannot eradicate it with pesticides or fungicides. It is just going to be there in the soil, and it pays to know that it's there in the soil and take that into consideration when you're choosing what kind of trees to plant. So we did some work on the humongous fungus. Alex and Nancy came out. She wrote Armillaria Crew Science Rules. We spent a week excavating trees. This is Alex, who I got several slides from, and Nancy, our coworker. And what we did was we went to the humongous fungus, and you can find, as I've showed you, mycelium under the bark of live trees, but it's actually, if you're trying to isolate the fungus, it's much easier to just find a small dead tree and rip it out of the ground, or cut a few roots and then rip it out of the ground. Then you have plenty of material to isolate the fungus from. We did that. Here's some of the root systems we pulled out of the ground. You can see not all the roots are rotten, but on some of them there are rotten roots. These are after we blew the soil off them, and basically to isolate, Alex cut a little bit of bark off and went for that white mycelium that was underneath, and put it in a Petri plate, and we kind of took over this bunkhouse and made it into a lab, and he isolated for 2 days straight from those root systems we'd ripped out of the ground. Then you've got an isolate in a plate that you can do things with. So how do you know if it's the same isolate or a different isolate? You do things called plate pairings. If they're the same isolate, it forms a reaction like you see on the right. They just totally overgrow each other. They're the same genetic organism, so they recognize that they're the same, so they just grow together. On the left is a plate pairing of two different genotypes, and there's vegetative compatibility genes. They recognize that they're vegetatively different, and they set up this zone line between the cultures. So this is before the days of DNA analysis. This is the way you determined if you had the same clone. When Brennan and the other authors did the work, they used this, and they also used DNA analysis, and confirmed their isolation of the genets with two methods. This is another one where these are the same, they overgrew, these are different, and they set up a barrier zone between the two. You can also run them on various kinds of gels, and these three that only have one band would be the same. These two that have two bands would be the same. This is kind of DNA fingerprinting in a way. This has been, this kind of look at the structure of genets or clones of a fungus has been looked at. This is work done in Switzerland by a friend of mine, Simone Prospero, and you can see this is 1 kilometer areas in a spruce forest, and you can see, and he did the same kind of work, plate pairing and DNA analysis, and you can see that these genets can occupy large areas. Now, sometimes they can overlap, and one isolate will be killing one tree and another one will be killing another one. They aren't exclusive necessarily, but you can see that they tend to occupy one area and then another genet occupies another area. All these little dots are dead trees that they had isolated from. So Armillaria does form large genets. This is the humongous fungus in Oregon. If you know Austin Junction, Prairie City is about 25 miles west of this view. John Day is about 35 miles west of this view. This is Austin Junction. And this is the large genet, genet D, that is the humongous fungus. This is GIS information from the original paper, and these are where Brennan and Catherine did their isolates, and then plate pairings, and you can see there's 6 identified genets. A couple of them were only one tree. but several of them were mini trees. And here's a close-up of the humongous fungus. You can see many trees had the same isolate in them. You'll see other dots on this map. This is the ones we recollected with Alex and Nancy in 2018. And you can see we collected some genotypes between the big genets that had previously been identified. And here's the results of the recent work. It basically, the white dots are the original isolates from Brennan Ferguson's work, and these other colored dots are isolates we got from our most recent work, and it basically completely confirms the identity of the humongous fungus. We only got one genotype from this whole area. So how big is this area? It's in the original paper, it's about 2,384 acres, which is about 3.7 square miles. It's very large. In the original paper, they did some estimates of its age, and the age of this large genet is estimated between, depending on the assumptions you make in genetic change, it's between 1,900 years or maybe as many as 8,500 years old. So it's been killing trees and moving in the forest since that time. I'll go back to this slide. You see this yellow outline. This was a large fire that happened after they did the original work. And much of the area outside the fire is old growth forest. Much of this has been either naturally regenerated or planted. And what's happening now is the young trees are starting to die. It's a disease of the site. The fungus was still in the ground, even though the trees above ground were burned. The root systems usually don't burn, and certainly by the time you get a foot deep in the soil, it isn't even heated up. So the fungus is still there. It's a disease of the site, and it's continuing to kill trees. Trees can have many, many contacts. This was actually an Armillaria study where we excavated an entire root of this old stump. And you can see how many roots cross this old root that's in the ground. If it's the same species, often these roots will actually graft to each other. Sometimes you see a cut tree in the woods that healed over. Well, it was grafted into a tree next to it, and it didn't even die when the tree was cut. It just kept living. So this is the reason this fungus can spread and occupy such large areas, is because of these roots go so far and the root contacts that happen within the ground. It's interesting, as I said, larix or larch often is not killed once it gets to be about 20 years old or so. But when it's young, it can be killed. And this is the proportion of the different species of the trees we collected in the Malheur that were dead. As you can see, it was easy to find dead grand fir. There just didn't happen to be many Doug fir there, but we did find dead ponderosa and lodgepole, and 20% of the trees were dead larch. So at a young age, larch is susceptible. So I've told you about the humongous fungus. You've heard a lot about its biology, and I believe I'm going to try to share my screen and we'll go to the video. And I'm sorry I don't have my chat up, but as soon as the video's over, it's a 7-minute video. I hope it works. It's pretty interesting. And as soon as the video's done, I'll go to the chat and try to address your questions. Are we good to go with this, Susan? And can you please tell me if you can't hear the sound once it starts?
Let's begin with a riddle. What living thing is bigger than a whale, yet hides out of sight? What could fill 250 semi-trucks, yet spreads itself thin? It's here, in the Blue Mountains of eastern Oregon, and it tries to suck the life out of whatever it touches. But to see it, you have to know what to look for.
People don't think of mushrooms killing trees.
This tree fights back. It tries to shove out an invading fungus with its own pitch, which oozes out of its bark. It's probably a lost cause.
The fungus will grow all the way around the base of the tree and then kill all the tissues.
Greg Phillips stands surrounded by infected trees, attacked by the honey mushroom.
So just about every tree in here probably has some degree of this.
He's a forest pathologist, a tree disease expert with the U.S. Forest Service. Trees, young and old in this stand, are dying in slow motion.
This is an older tree, yes. It's hard to tell how long it's been infected. It could be 20, 30, 50 years maybe before it finally dies.
If you're thinking of a classic mushroom with a cap and gills and spores, well, yes, the honey mushroom does look like that. For a couple of weeks each fall, it erupts in clusters, sometimes right through a tree's bark. But most of the year, it's just a thin white layer that packs a lethal punch.
This white material here, it's the actual fungus, and you could peel it off like latex paint.
That white fungus spreads up under the tree's bark, rots its roots, and proceeds to both starve and dehydrate the tree.
And then there's no movement of water or nutrients up and down the tree when that happens.
Greg and fellow researchers brought us into the Malheur National Forest because this is the spot that put Oregon on the Humongous Fungus map.
I was the one that raised the alarm, saying this was different.
Greg Whipple first noticed the fungus in 1988. It seemed to cover about 400 acres.
And in '88, that was much, much denser up on that knob, and they have since died from the root fungus.
Now we know the footprint of the fungus is far larger than a few hundred acres.
It covers, in this particular area, over 3 square miles. We haven't seen anything else in the literature that would suggest that anything else in the world is larger in acreage.
They've dug out samples far and wide across this section of the National Forest. In every spot across thousands of acres, they find more of the fungus. It's not just the same type of fungus. They're convinced this is the largest single living organism in the world.
We know that because we took samples of the fungus, brought them back to the lab, and then identified them using DNA analysis and other techniques to tell that it was all the same organism.
If you could scoop it all up and pile it together, scientists calculate it could weigh at least 7,500 tons, and maybe up to 35,000. That's the weight of more than 200 gray whales. It's also not new. The humongous fungus is not something you can blame humans for introducing.
When you realize that this fungus spreads at about 1 to 3 feet a year, and you have something that large, you can calculate the age. And we're looking at anything from 2,000 to 8,000 years old.
Its scientific name is Armillaria. It may be easier to remember popular names, and there are several: honey mushroom, humongous fungus, shoestring fungus. Retired pathologist Craig Schmidt says that last nickname describes yet another form this fungus takes when it sets out to spread.
They look just like fine roots. These actually grow through the soil and help spread the fungus from one tree to another. This is one of the reasons why Armillaria has been called a shoestring fungus, because of these structures.
With its underground tentacles, it spreads out in all directions. A forest of interconnected roots feed the trees, but also provide an unwitting pathway for a parasite on the prowl.
It doesn't just exist here. This is the largest one, but it's throughout the world, and it does similar things in forests throughout the world.
You might guess this could pose big trouble to timber companies, and it has.
I'm looking for new dead trees.
Near Glenwood, Washington, researchers have been experimenting with various ways to handle the honey mushroom for 40 years.
This is private timberland. A lot of the research we do is very cooperative in nature. They have a lot of questions. We at the state have a lot of questions.
Dan Amdahl and Amy Ramsey are pathologists with the Washington State Department of Natural Resources. In the 70s, a team set out to see if they could eliminate the fungus entirely in this forest. They not only cut trees, they dug out the stumps. In some areas, they went farther and raked out every last fibrous root they could find.
We significantly reduced the amount of fungus that was in the soil, and so subsequently, more pine trees survived on those sites. The challenge is it's very expensive and oftentimes prohibitive to do that level of intervention.
Timber companies can't afford to dig every trace of it out, so researchers are trying to learn something else. What's the best tree to plant that can manage to live with the fungus?
We have ponderosa pine, we have a western larch, we have a Douglas fir, and we have a white pine. We chose this site because the fungus is living on this stump. Armillaria is an aggressive pathogen. It's the baddest fungus on the block in this particular neck of the woods. And so we're looking for a tree that can grow in its presence.
The results might ultimately be able to tell timber companies, plant something else.
Oftentimes it's foolish to plant the same species in a site where you just harvested in areas that are infested by the disease.
There's another way to view Armillaria. It's simply nature helping nature.
Well, if you look at it from an ecological perspective, it's actually helping recycle the trees. Kills them, rots them, they go back into the soil. There's a wildlife benefit to these trees. Once they're dead, they decay, the birds begin to excavate them and use them for cavities.
The humongous fungus lurking underfoot is here to stay.
We learn to live with it. Then realizing we're dealing with an organism that is in the thousands of acres in size and in the thousands of years in age, that's pretty overwhelming. And then to realize as a forester that I'm not going to have much effect on that. It's part of the landscape.
Yeah, that was great.
So that's all I had. I hope you enjoyed it. I'm gonna look at the chat window and see if there's anything okay, the term genet, I see from Susan. I don't know the term genet. It's basically clone. A genet is genetically the same. So if I was to make cuttings of my rose bush and give you one, those would be genets. They're genetically identical. Are there multiple areas on the map related genetically, or just the largest one considered in the calculation? It's that genet D or D that I showed you is the largest one. There's several others, as you saw in the maps, that are close by that are just about as big. But yeah, Dan Amdahl and Amy, I'm glad they got in there, because that area at Glenwood was previously published as the biggest living organism. And that was based on some work that Greg Phillip, who was in the video talking, he's retired now. He was the regional pathologist, but he did some work when he was in graduate school, and he previously claimed the humongous fungus, and then he moved over to LeGrand and they found the bigger one. And there may well be bigger ones out there. It would just take doing the collecting and doing the plate pairings to see. You know, you can find, and the biggest fungus may not be Armillaria. It may be something else, but this is backed up by good, solid research, and it just shows you how extensive it can be and how many different ways you can look at it. It's either a tree killer, or it's a good edible, or it's recycling wood into the forest. You know, it depends, as we always say in our business, it depends on your objective for the land, whether it's a friend or foe. So, yeah, I have eaten it. I had a friend in another pathologist years ago that lived in Colville, Washington, and she collected so much Armillaria and dried it. She traded it to all her friends for all her winter vegetables, onions and potatoes and everything else. It is a good fungus when you can find it fruiting. Is it bad?
Yeah.
I'm just going down through the. Is it bad to bring home honey mushrooms? Well, no, but I wouldn't throw the leftovers out in my garden either. As I showed you in that pear orchard, it can cause mortality in many hardwoods and conifers. In fact, in the Willamette Valley, in the edges of where the Willamette River used to flood, there's lots of buried wood. Well, there's lots of Armillaria. And in fact, they were having problems growing raspberries because Armillaria was infecting the raspberries. So it has a very wide host range. And if you bring any home, I'd eat them. And I don't think I'd put them in the compost heap. The Utah aspen grove is not fungus, it's aspen. It's a large clone of aspen that has also reproduced vegetatively. If you burn an aspen grove, every root will sprout little aspens. And in fact, in Colorado especially, or places where they've got large aspen groves, you can see in the fall, one clone will be turning color and the one right next to it is still green. So you can almost identify them on the landscape like that. So good. I've given you a totally new view of fungus and what it does in the forest. That's sort of what I intended to do. How is this affected by fire? Almost not at all. It'll kill the trees, but most fires do not heat the soil up very much. And unless the root system is very decayed, usually it doesn't even burn out a root system. So a fire might reduce the hosts for a little bit, but it's not going to kill out the fungus. So could red belt polypore be a contender for world's largest fungus? I have a picture of one I found on a live tree in Alaska, and it's by far the biggest conch I've ever seen. And, you know, there's Nobilissimus, there's some other large conchs, but certainly red belt is among the contenders. And I kind of showed that to you because it's something people see in the woods commonly, but sort of don't know its role, and now you know what it does out there. Yeah, the Utah aspen grove, it's just 116 acres. How could it be the biggest? So the Utah aspen are supposed to be the largest biomass by weight. You know, I doubt it, but it's certainly possible. So that's about what I've